the notes. I don't think we talked about... Did we talk about the bar body? Or the sex-linked genes? Right about here looks about right to me, where we left off. So everybody was able to find this document okay on Canvas? Okay. Okay, now we're recording again. So with the sex-linked genes, these are going to be ones that are on the X and Y chromosome. Okay, now we're recording again. So with the sex-linked genes, these are going to be ones that are on the X and Y chromosome in there, which are your gametes or your sex-determining chromosomes. And so with these, you have a short segment at either end of the Y chromosome that is going to be homologous with the X chromosome, but only a small portion of that is going to be homologous because there is a significant size difference in there. So for males, they're going to be X and Y. Females are going to have 2 X chromosomes in there. You get one from each parent. The sperm is going to determine the sex because you're either going to get an X or a Y sperm. The mother only produces X chromosomes, and that's what all of the eggs will have, but the sperm will have the X or Y, and that's why the male determines the sex of the infant in there. So in humans, the signs of your sexual development are going to begin to appear at about 2 months in there. It's sometimes, some sources are going to say 5 or 6 weeks in there. Prior to that, your sexual development is generic. A lot of sources will say by default you're developing as a female because unless you get a signal to change, by default it develops as a female in there. So in 1990 they identified this gene, the SRY gene, the sex determining region of Y is what it stands for, on the Y chromosome that is required for testes development in there. If there is no SRY gene, regardless of what the rest of the chromosomes are, it is going to develop as a female in there. So the Y chromosome, there are 78 genes. It codes for about 25 proteins, and only about half of those are expressed in the testes in there. With crossing over, you can occasionally end up with the SRY gene crossing over onto the X chromosome. Statistically, it's about 1 in 10,000 normal appearing males are actually chromosomally a female, and 1 in 10,000 normally appearing females are actually chromosomally a male, and you just don't know till you have no fertility in there or you don't have the proper sexual development in there. So like you wouldn't go on if you were, you wouldn't be able to be fertile because you don't have the correct gamete pairs to make in there. So sex-linked genes are those located on either of the sex chromosomes. The Y-linked genes are going to be on the Y chromosome. They're going to be passed from the father to the son. No women get any Y-linked disorders in there. There's very few genes that are on there. That chromosome has so few genes that there is very few Y-linked disorders in there because of it. On the X chromosome, you've got about 1,100 genes on the X chromosome, and so an X-linked gene will be present on there. Since males and females are going to have a different pattern of inheritance on here, there's a different number of chromosomes than the autosomal genes on these. So the way they will show this on here is that if you've got a parent, male is XY, female is XX. If you have trait N on there, you can see they've put it up as a superscript on there. When they do the Punnett squares on there, it will show the different patterns of inheritance on there, because for a male, you may or may not inherit it. For the female, they're always going to get an X chromosome, and you may or may not inherit it on there as well. So the X chromosome has many genes that are unrelated to sex in there. A father's going to pass his X-linked genes to all of his daughters because they're going to be on his X chromosome in there. None of their sons, because the sons don't get the X chromosome. The mother can pass X-linked alleles to both daughters and sons. X-linked recessive traits are only going to be expressed if the female is homologous. She has to get both recessive genes in order for it to be expressed. For males, they only have one locus, only one gene in there. So if they receive the recessive allele on there, they're going to express it because they don't have anything else to override it in there. So more males are going to have X-linked disorders than females will, because females have the chance of having another chromosome to override it. Color blindness is one of those things that's almost always inherited as an X-linked disorder. So you're going to see that far more often in males than in females. Another thing that is X-linked is male pattern baldness in there. Again, you'll see it more often in males than in females, although you can have both of those things occur in a female. So Duchenne's muscular dystrophy is also another X-linked disease in there. About 1 in 3,500 males in the U.S. It results in progressive weakening and loss of muscle coordination in there. These will rarely live past their early 20s because they are missing a protein called dystrophin in there. How this will not necessarily get passed on as much, because if they are not going to live past their early 20s in there, a lot of times they don't reproduce. So the younger a disorder kills someone, the less likely they are to pass it on in there. Hemophilia, it's an X-linked recessive trait. So the person is missing one or more of the proteins needed to clot blood. So since females get 2 X chromosomes in there, you're not really going to have 2 X chromosomes acting in all of the cells all the time. So instead, one of them is going to become inactivated during embryological development in there, and it becomes a bar body in there. It just becomes a little bar in there. It's not gone, it's going to fold itself up and be inactive in there. It's random which chromosome is going to turn into the bar body, and it's going to be independent in each embryonic cell at the time of X inactivation. So a female may have 1 X chromosome from one parent inactivated in one cell and a different one inactivated in another cell. So she could have a mosaic pattern of the 2 cell types in there. After X inactivation, all of the mitotic descendants are going to have the same inactive X gene in there. And it's going to involve modification of the DNA and the histone proteins bound to it. They're going to attach methyl groups to it, to the nitrogenous bases. The X inactive specific transcript is going to become active only on the chromosome that will become the bar body. So that's going to be a set of directions that's going to tell it how to turn that into a bar body. So when we talk about linked genes tend to be inherited together because they are located near each other on the same chromosome. So now we know with crossing over that you're going to have, on average, about 3 sections cross over on the chromosome in there. So when you're looking at which sections cross over, the closer they are together, the more likely they're going to be involved in the same chunk that does the crossing over in there. And so that's what we refer to as a linked gene. They're close to each other, and when chunks of it cross over, they're more likely to go together in there. So traits are usually inherited in specific parental combinations because genes for characters are near each other on the same chromosome. So the closer things are, the more on the chromosome, the more likely you are to pick up those traits from the same parent in there, just because of the likelihood of the crossing over occurring together in there. So the genetic recombination is the production of the offspring with all of the combinations of traits from either parent in there. So how we get this genetic recombination in there, you're going to have recombination of unlinked genes. This will occur by independent assortment on the chromosomes in there. So you have the phenotype of the parent in the P generation, and then the recombinants or the recombinant type is the new combinations of phenotypes that you have in the offspring. And so there's a 50% frequency of recombination observed for any two genes on different chromosomes. The reason they state that with different chromosomes is if they're on different chromosomes, they can't be linked. It's completely random on each chromosome. Are you going to get mom's or dad's version in there? With linked genes where you have the crossing over, two genes on the same chromosome of the same parental type are considered linked if there is a frequency greater than 50%. So if you get a set of genes paired together and they're more than 50% of the time, they're going to say it's linked because it came in the same section in there. So the crossing over accounts for the recombination in linked genes. It occurs while the replicated homologous chromosomes are paired during prophase of meiosis 1. And there's going to be a set of proteins in there that are going to orchestrate this exchange of the corresponding segments from one parent to the other parent's chromatid in there. So these new combinations of alleles, what this does is it provides more variation for natural selection to be able to have the stronger one thrive in there. You can't really have natural selection if everybody gets the same traits. There isn't going to be one that's better that survives. The only way we're going to know which one is better is to throw those variations out there and see how they do. So the crossing over, recombinant chromosomes, and random fertilization all help to increase genetic variation and provides more opportunity for natural selection to work. So this just shows in here how you can have sections in here that will trade off for your crossing over in the chromosomes. And you can calculate out the recombination frequency if you really wanted to. So genetic maps are going to be ordered lists of the genetic loci along a particular chromosome. So this was one of the first ways that they actually started to map things, is they would take a chromosome and we'd say, well, we know this gene is located here relative to this gene. So it would give you an idea of the order the genes are in on the chromosomes. The recombination frequency is the percentage of the recombinant offspring, and it's going to depend on the distance between the genes of the chromosomes. So this was part of how we learned about the linked genes, as we were seeing that some of these that were closer together were occurring more together in there. So part of this assumes that crossing over is random, and Alfred Sturtevant actually proposed the idea that the further apart two genes are, the higher the probability that a crossover will occur between them and get more recombinant frequency in there. So another type of map that we can do are linkage maps. And this is a genetic map based on the recombination frequencies. So the map units are the distance between genes. So 1 unit is equivalent to 1% recombination frequency. So when you take the information from 2 of those maps, you can look and see, okay, this is the order we know they're in, and this is the relative distance of them based on how often they will recombine in there. So with these types of maps, despite being on the same chromosome and physically connected, the genes are going to be genetically unlinked and the sort independently. And linkage maps are based strictly upon recombination frequencies. So they will just give you one limited type of information. So another type of map we can make in this are the cytogenic maps, and these are going to locate the genes with respect to the chromosomal features. So this will help us be able to find the linear order of the genes, but the spacing between them is not going to be identical in all maps in there. So what if we alter the number of chromosomes? This is not going to be a good thing for any living thing to alter the number of chromosomes. That is something that we're pretty sensitive to. There's a very low survival rate with it. Large-scale chromosomal alterations in humans and mammals usually result in spontaneous abortion. And those who do survive are commonly going to have developmental disorders in there. So if you have a non-disjunction, this is where members of homologous chromosomes don't move apart properly during meiosis 1, or the sister chromatids fail to separate during meiosis 2. That's how you can end up with the extra number in there. So this is showing as we go. Hello? He is not in this room during the day. He's in here in the evenings on Tuesday and Thursday. Do you need to deliver them, or you're... I can give you his phone number. That's okay. I know I have his phone number in here somewhere.
Well, I can get that for Teresa. That's all right. I'll have her. Are you Wendy?
Yep.
Thank you.
Now I can put a name to the face in there.
I need to know if he was perfect being tested.
I don't think it really matters
Then I'll
on
put
there.
the easy one in. How's that?
I'll put his phone number on there for you.
Thank you, Wendy. Sorry for the interruption.
No problem. Okay, so this just shows here where we have this one lost a chromosome because this one gained an extra one. So you have 3 in here. We call it n plus 1. Here, n minus 1. N represents the normal number, so you have the normal number plus 1, the normal number minus 1. When you have the non-disjunction up higher in there, you get 2 of them that have that. Two of them that have an extra 2 that are missing 1. If you have the non-disjunction later in meiosis 2, you get 1 that has too many, 1 that has too few, and 2 that are normal. And the non-disjunction is not a normal thing to happen in there, so fortunately this does not happen very often. So aneuploidy is going to be the zygote that has an abnormal number of a particular chromosome. So the monosomic are going to be fertilization involving a gamete that has no copy of a particular chromosome, so it's missing 1. This would be your monosomic one. And the trisomic are going to have an extra 1, so these are your trisomic ones that have 3 in there. So polyploidy is the organism that has 2 or more complete chromosome, an organism with more than 2 complete chromosome sets in all of their somatic cells. This is a lot more common in plants. Plants can tolerate this kind of thing a lot better than humans can. Some plants will get extras in there, but in the animal world, this just does not work well. So how can we alter the chromosome structure in here? Deletion occurs when a fragment is lost or deleted. And it can be lost and just go into the cell and be reabsorbed somewhere else, or it can end up attaching as an extra segment on a sister chromatid in there. Neither one are going to be good. A duplication is going to be that deleted fragment attached to a sister chromatid. An inversion is going to be where a chromosome fragment reattaches to the original chromosome, but in the reverse direction. So you take the instructions out, you flip them around and stick them in backwards. They don't read the same when you put them in backwards, so it puts in something else. And then translocation is going to be when a fragment joins a non-homologous chromosome. And translocation can occur from various other causes in there. So in a male, if you get large deletions in the X chromosome, it will tend to be lethal because they only get one chromosome in there. The female has a little bit better chance of surviving that because she does have the two X chromosomes in there. And with that mosaic pattern in there where you have some of them, you can assume that sometimes that problem chromosome is going to turn into the bar body, and sometimes the normal chromosome will turn into the bar body. There can be some normal cells in there that may allow things to function a lot better and not be lethal in there. So the human disorders due to chromosomal alterations. So Down syndrome or trisomy 21. This occurs in 1 in 700 children in the U.S. There is an extra chromosome 21. They will tend to have pretty characteristic facial features that will go with it in there. Short in stature. They'll tend to have kind of broad space between the eyes and a smaller nose in there with their facial features. They'll have heart defects that a lot of times can be surgically corrected in there. Some developmental delays. They have quite an increased chance of leukemia in there that will go with that. They also have a greater risk of Alzheimer's at an earlier age in there, so Alzheimer's can occur far earlier in somebody with trisomy 21. But they have decreased risk of high blood pressure and atherosclerosis. There is increased frequency of trisomy 21 due to maternal age in there. Most of the chromosomal disorders are going to increase in frequency with the mother's age in there. You do see a little bit of increase go along with the male age in there, but you tend to see other disorders increase with paternal age. It's the chromosomal ones with maternal age in there. And most of these are due to non-disjunction during meiosis 1 in there. The aneuploidy of the sex chromosomes, these appear to have less effect than in autosomes. Because the Y chromosome has relatively few genes on there, that can help to minimize the actual visible effects of it. And then extra X chromosomes that can be inactivated as bar bodies in the somatic cells help to cushion the effects in there. So with Klinefelters, this is going to be XXY. This would occur in about 1 in 500 to 1,000 live male births. The male is going to have male sex organs. The testes will tend to be small. They will usually be sterile in there. An extra X chromosome gets inactivated, but a lot of times you'll see some breast enlargement in there and female body characteristics. And they'll have subnormal intelligence, so more developmental problems that will go with this. With XYY, this one is sometimes referred to as the super male. That occurs in 1 in 1,000 males. They have normal sexual development and are fertile in there. They're taller than average. There's been some interesting studies that they've done with the XYY in there. There was, for a while, they were saying that there was, because of potential increase in testosterone levels, they were more prone to violence. And what I found really disturbing was the study that criticized that said they only beat their wives. And I'm like, okay, so that's supposed to be okay. But there is some disagreement in there with that, but there has been some interesting studies out there that you can, if you're interested in it, you can look for yourself in there. Anytime you look at studies, there's going to be disagreement most of the time. And it's just interesting what they have found with some of these. The XXX female, this is about 1 in 1,000 females. Healthy, usually no features other than being taller than average, but an increased risk for learning disabilities. They're usually fertile. The more extra X chromosomes you get, usually the more severe the learning disabilities get with these. With Turner syndrome, monosomy X, this is 1 in 2,500 female births. This is the only known viable monosomy in humans. And phenotypically they're female, but they're sterile because they don't have the second chromosome to put into their gametes. Their sex organs do not mature. A female with Turner syndrome can develop secondary sex characteristics if they're given estrogen therapy. They'll have normal intelligence. A lot of times that estrogen therapy is going to come a little bit late because you're also relying on a parent to look and say, wow, my daughter is 16 years old and has no signs of puberty. Maybe we should do something about it. So a lot of parents there's going to be a delay in there, and not all parents will actually do anything. So the closer to the normal time they can do the estrogen therapy, the better for that person in there to get them on a normal track. The longer it's delayed, you're going to have psychological factors that are going to go in with going all the way through high school looking like you're 8 in there. So some structurally altered chromosomes. So this has a French pronunciation to it that I butcher. It's cri du chat in there. It means cry of the cat. And there is a specific deletion on chromosome 5. So these people are going to be severely intellectually disabled. They'll usually have a small head and facial features in there. And they will maybe make it into being a young child, but most of them will die as infants in there. But their cry sounds more like a distressed cat with more of that yowling type of sound than a normal infant's cry. So CML, the chronic myelonic leukemia. Chromosomal translocations are implicated in certain types of cancers, and this is one of them in there, including this. It's a reciprocal translation during mitosis of the cells that will become your white blood cells. A large portion of chromosome 22 gets exchanged with a smaller fragment from the tip of chromosome 9, and it produces what they call a Philadelphia chromosome. And it's going to be a much shortened chromosome 22, but what is missing in there leads to uncontrolled cell cycle progression, which is why you have in a cancer in there that the cell cycle is thrown off. So some of the inheritance patterns are going to be exceptions to the standard Mendelian inheritance patterns in there. And a lot of these are things that we have really just started to understand in the last 10 years in these. Genomic imprinting, this is going to be a variation in the phenotype depending on whether the allele is inherited from a male or the female parent. And a lot of times what's going to cause the change is a methyl group is added to the cytosine on one of the alleles. So it's just a carbon and 3 hydrogens added on to one of those base pairs in there. This will occur during the gamete formation, and what it ends up doing is it silences one of the alleles of the certain gene so that it will be imprinted differently in the sperm and egg. So the zygote only expresses the allele of the imprinted gene that's inherited from the male or the female parent in there. Old imprints will erase in each generation, and each new gamete imprint will be according to the sex of the individual forming the gamete in there. And it's thought this only affects a very small portion of the mammal genome. Most of the imprinted genes are critical for embryological development in there, so they're going to have a role early on, long before you're even born in there. So it's showing it being studied in a mouse here. So the normal size mouse, And with getting this one mutant in there, you get the dwarf mouse. And I was not able to find anything for an example of a specific one in a human. So inheritance of organelle genes. So you do have genes outside the nucleus. Most of your genetic information is in the nucleus, but you've got these few that are outside. In animals, the mitochondria are going to have them. The chloroplasts and the plastids will have them in plants, and they're just small circular DNAs with some genes on there. So the organelles reproduce themselves and transmit their genes to the daughter organelles. They're not going to follow the Mendelian inheritance because organelles just make a copy of themselves, and they're like little clones of themselves within the cell. In most plants and animals, the zygote is going to receive the cytoplasm from the egg. So when you have fertilization occur, you got the big old plump egg that's there. The sperm comes along. The sperm basically drops off the genetic information, and that's it. What you have in your cytoplasm all came from your mom in there. So your mitochondrial genes in the animal world generally are all going to be inherited from your mother. So you inherit a little bit more from mom than you do from dad. So most of your mitochondrial genes are going to make up the protein complexes of the electron transport chain and ATP synthase in there. So more of the metabolism in there, slightly more of your metabolism characteristics in the cell can be inherited from mom. So if mom is really athletic, has lots of energy, that's a good thing for you. Or like in my case, if mom is a hefty couch potato, you get to fight that the rest of your life. So with these genes in there, defects reduce the amount of ATP the cell can make, and so the muscular system and nervous system are susceptible to disease in there. When I was reading on this, with the mitochondrial mutations in there, they figure it will contribute to at least some cases of diabetes because of it affecting the metabolism, heart disease, which a lot of heart disease is related to dietary issues and the metabolism, and then Alzheimer's in there. A couple other conditions that are going to be carried in those mitochondrial DNA, myocardial myopathy, where you'll have muscle weakness, intolerance of exercise, and muscle deterioration. And Leber's hereditary optic neuropathy. This can produce sudden blindness in somebody in their 20s or 30s. There's 4 mutations that cause this that will affect oxidative phosphorylation in there. Basically, you affect energy getting to the optic nerve in there, and you get a neuropathy that does not allow that nerve to work and carry the visual impulse anymore. So on to chapter 16. Let's go ahead and we'll take a break for a couple minutes. So in this section we start to look at the structure. of DNA. And the model of DNA, with it being the double helix shape, was come up with in 1953 by Watson and Crick. There's actually a lot of videos out there. In our library there is a video series on DNA and chromosomes and how they came up with a lot of the stuff from the Howard Hughes Medical Institute that has a lot of historical stuff. If you prefer to see things in like a video format, it's something you can sit down and watch. And this stuff is still relatively new, where Watson and Crick are dead, but a lot of the other people who have worked on the stuff with the Human Genome Project, like Craig Venter, is still alive and still doing research out there and working on things that it's kind of cool to be able to actually see videos that have these actual people that have come up with these things on there, coming up with it in there. I think Craig Venter is kind of interesting in the videos. He's one of those people that I think he's kind of got a little too much energy for his size, but you know, that's the kind of person it takes to do that kind of work too, in there. So they're available in our library. They won't let you take them home, but you can always watch them in there. I think they're actually on DVDs, but they've got whatever player you need to watch it in the library if you're interested. So the genetic material. So a researcher, Morgan, showed that genes were part of the chromosomes. And at that point, they knew they were genes, but they didn't really know, okay, what's on these chromosomes and what are these genes made out of? And so once they started to figure out that these genes were the units of hereditary and things like that, that's when they decided, okay, the DNA and the protein are possible candidates to be this genetic material that actually is carrying this information. So the role of DNA in heredity worked out while studying bacteria. They looked at bacterial transformation in there. What happened is they had bacteria that were basically put in a surrounding or an environment that had pieces of DNA in it. And when that bacteria took up the pieces of DNA into their own cells, it started to manifest some of the traits that were in that DNA. So that gave them the information to say, okay, the DNA is what's carrying these instructions in there. So transformation is a change in the genotype and phenotype due to assimilation of external DNA by the cell. There is multiple ways that a cell can be transformed in there. So it can be transformed by simply it just comes into the cell through the cell wall in there. It can be transformed by a virus that does that. So the term transformed is pretty general in there. It just means you've taken in DNA that wasn't originally there in there. So there was the evidence that DNA can transform bacteria in there by those cells that were put into that environment with the DNA. And there was also the evidence that viral DNA can program cells through studying bacteriophages or phages, which are just viruses that infect bacteria. A virus is basically DNA or RNA enclosed by a protein coat. It doesn't have any organelles or anything else like that. They're pretty simple objects in there. And in order for the virus to be able to do anything, it has to go into a host cell and use that host cell's machinery. So that gave more evidence that the instructions were in the DNA, because that's basically what the virus put in. And then they were able to look and see, okay, we have the proteins that the virus was wrapped up in are left outside the cells, and it was just the DNA or RNA in either case that went in to say it was the DNA or RNA holding the instructions instead of the proteins in there. So they studied the T2 phage that infected E. coli. They had these 7 phages in there that were called T1 through T7 in there that they studied that would affect E. coli in there. And they used these radioactive proteins and found that when the phage DNA entered the cell, the radioactive proteins were left on the outside. And when that DNA went into the cell started taking on the directions of that DNA, and the proteins were still on the outside to distinguish that was the genetic material in the DNA. So additional evidence came from Chargaff's study. He studied the ratio of nucleotide bases and found that the number of A's you have is approximately equal to the number of T's. So adenines is equal to thymines, because in DNA, adenine is always going to pair with thymine. They fit like a lock and key together. So you should have the same number of them. And then found that the number of C's, which are cytosines, is approximately equal to the number of G's, which makes sense since those two will pair together in there. And what he found was the base composition varies between species. So everybody's going to have A, T, G, and C, but the ratio of A and T versus G and C will vary in there. And within a species, the number of A and T bases are equal and the number of G and C bases are equal. So when we make this structural model of DNA, we have a double helix shape in there, and it was determined using X-ray crystallography. And the strands are going to run anti-parallel in there. So you've got your 2 sugar phosphate backbones. And a long time ago when we talked about the chemical structure of this, we talked about there being a 5' end and a 3' end. What that means is you're going to have 5' at one end, 3' at the other. Then you have 5' to 3', and they line up like this. So they're running in opposite directions versus like this. And with the size of it, the helix makes 1 full turn every 3.4 nanometers. And the bases are stacked 0.3 nanometers apart. So there's going to be 10 layers of base pairs for every turn. So by the time you go all the way around once, there's 10 base pairs that make up that turn. And they are on here. And so I'm going to, for being able to practice and kind of play with the DNA model, I've got a model that I'm almost done making at home with magnets that you can put on the board and little instructions that says, okay, do this, label this part on here. And they fit like puzzle pieces. So I'll have those ready. I'll hopefully get them finished up tonight and can bring them in so that you're welcome to come in and play with them outside of class time. And I can also give you the printouts if you want to make your own. However, those magnet sheets are kind of expensive. So this is showing the double helix in here, and then you've got your base pairs that are coming across. This is showing the base pairs in here where you've got your G and C, A and T pairing together. And so it's 3.4 nanometers for 1 whole turn. So it's the 0.34 between each of these spaces in there. So DNA is going to have proteins in there that help with the replication and repair. We use a semi-conservative model to make copies of the DNA. So if you have this original strand here, what's going to happen is it separates, and you can only have the complementary piece fit with it. Just like you can only put your house key in your door, you can only put A with T in here. So when you separate it, it's going to make a new strand in here, which is the light blue one, that's going to match it. So now you end up with 2 strands, and we call it semi-conservative because each of these new strands has half of one of the old ones still in it, in there. And by doing that, it's going to help to ensure that the copy is correct because you can't fit anything else with A except the T in the DNA. So your parental molecule has 2 complementary strands. They're going to be held together with hydrogen bonds in there with their specific partner. So your first step is to separate those 2 strands and then you put the complementary strands in there. So when we go to do this and we get started in there, we start out, our DNA is double-stranded in there. And we're going to look at it first in a round bacterial chromosome. You're going to have this origin replication where you're going to begin to initiate this process, and you're going to form kind of a bubble in between the 2 strands that you can see right there where there is a bubble in there where you're separating them. So what you get is on either end of the bubble you have an origin of replication in there. So it can spread in 2 directions out from those 2 origins of replications and fill it in to make 2 new rings that will come from that. So the bacterial chromosomes being circular, you have that one origin that's going to make 2 origins to spread. The eukaryotic chromosomes, being that they are linear in there, you're going to have this start in multiple places. So if you just have 1 chromosome, it doesn't take that long to copy it in there. If you did 1 at a time with 46 chromosomes, you're going to be waiting a while for that to all happen. So ours has multiple places that it will split and create these origin or replications, so you can have it going on simultaneously in multiple places. And that's us that only have 46 chromosomes. There are organisms out there that have hundreds of thousands, even millions of chromosomes. in there. And if you had to wait for each one to go individually, they'd never get anything done. So it's showing in the eukaryotic that you make multiple of these bubbles, and the bubbles start to spread together in there. So the replication fork is going to be the Y-shaped region. So you have kind of a Y shape here where this is spreading. So you can see it makes a Y in there at the end of the replication bubble where it's being unwound. So everything gets to have enzymes that control it. Helicases are going to be the enzymes that unwind the double helix at the replication fork for the two strands. So it's kind of like if you are trying to peel something apart that's all twisted like this. If you untwist it, so it's like this, it's a lot easier to separate it, your bubble, than it is if it's tied up all over itself in there. You've got these single-stranded binding proteins that are going to bind the unpaired DNA strands to keep them from repairing. So the DNA wants to close back up and you stick something in there. It's kind of like if you change a bike tire in there and you get it off of the lip in there and you're trying to pull around on the sides of it. You can work away at the two areas, but you stick in your tire iron so that it doesn't keep popping back onto your wheel. It's the same thing with the DNA, only doesn't take as much muscle to do it. A topoisomerase is going to help to relieve the strain ahead of the replication fork. It's going to start to break some of the bonds, swiveling and rejoining the DNA strands at the end, so that you don't just continue to have that coil get tighter as you are separating it. You can actually have it uncoil to release some of the tension. The enzymes that synthesize DNA cannot initiate the process. They can only add nucleotides to the end of the existing chain that is base paired with the template strand in there. So since they can't initiate the process, that means you get to have a whole nother set of enzymes that initiate the process and a whole bunch more regulation by having more enzymes in there to regulate what DNA is being copied and which is not. So at the beginning of it, you're going to have a primer, which is RNA strands in there of initial nucleotides that are going to be produced during the DNA synthesis. This primer is going to be the region where you can get things started, and some of the enzymes are only going to recognize that RNA primer. They're not going to actually recognize the DNA. It's going to help so that you know where you have that primer in there, you know you're starting at the beginning of the sequence you need to copy, not just someplace randomly in there. So the primase is the enzyme that's going to synthesize the primer and starts the complementary RNA chain from a single-stranded RNA copy. So it's going to add one nucleotide at a time using that template strand in there, and that primer is usually going to be 5 to 10 nucleotides long, and it starts from the 3' end of the primer in there. So when you synthesize a new strand in there, we have these DNA polymerase enzymes in there. So a polymerase means that it's going to make a polymer in there, and the ACE just means it's the enzyme that facilitates that. And it's going to do this by adding new DNA nucleotides to a pre-existing chain. In here, I have referred to it usually as just DNA polymerase or RNA polymerase. There are different variations in there. In the last 15 years, they have identified that eukaryotes have at least 11 different DNA polymerases. They're likely not done finding them all. And the process is going to be essentially the same in there without beating you up knowing names of individual polymerases that work under certain circumstances in there. Right now, we don't understand them well enough to make it worthwhile to memorize the differences in there. Most DNA polymerases are going to require a primer and a DNA template strand. There are RNA polymerases, so just be aware when you look at the name in there, those two are going to look alike. DNA polymerase means you're adding on more pieces of DNA, your DNA base pairs. RNA polymerase means you're adding on your RNA base pairs, which are slightly different. So it's going to add nucleotides to the RNA primer at a rate of about 500 nucleotides per second in bacteria, and at a rate of about 50 per second in human cells. So it's quite a bit slower in the human cells for this. Each nucleotide added is going to come from a nucleoside triphosphate in there, and as that monomer nucleotide is added on, you're going to have a little bit of energy spent in there that's going to drive the polymerization in there. So just like when you have to spend energy to build muscle, you get to spend a little bit of energy to build the new DNA strand. They are going to parallel, or they're going to elongate in anti-parallel directions. So the strands run anti-parallel to each other in opposite directions. And so replication is also going to be in opposite directions in there. The DNA polymerase can only add to the 3' end of the primer or the growing strand of DNA. So it's going to elongate in the 5' to 3' direction. So if we have this 5' here and this is 3' over here, it only adds on to this end. When you have one enzyme working on two strands that are going in opposite directions, that creates kind of a weird scenario where we have a leading strand and a lagging strand. So they run like this. So when this is opened up, and we have split it open like this, you have 5', 3', and when you're filling in new nucleotides along the way here, you're going along like this. So you're adding on to the 3' end here. And this works good because as this splits open on this one, you just keep adding on here as it's splitting open. However, on this side, you've got the enzyme in the same place. What you have to do is go part way in there, or you're going to have to start and go here out, here out, here out, like this. So this one that gets to go in a nice normal order is going to be your leading strand. This one that has to do in little chunks because you can only add on to this 3' end is considered your lagging strand in there. And then you make each of those little pieces in there. So your lagging strand is each DNA polymerase must work in the direction away from the replication fork in there. And therefore you make these little segments that are usually 1,000 to 2,000 nucleotides long in E. coli. In eukaryotes, they're a little bit smaller. They're 100 to 200 nucleotides long. And we call those little fragments Okazaki fragments. And so afterwards, we have to glue those little Okazaki fragments together, and DNA ligase is going to come along and do that so you can have it be one solid strand in there. So this picture here shows it where you've got your replication fork, and this is going along in here where you get to have it all be one nice smooth process. And with this one, you get to do here back, here back, here back, and move here back like that. So you do it in the little chunks. And so this is showing where you have these Okazaki fragments that get made in the blue. And then you're going to have to glue them together. After you glue them together, then you come along and you clean up the extra sections in there so that you get a nice overall clean piece of DNA in there. So you have the DNA replicating machine, which is going to be the DNA that's being replicated and all of the proteins in the complex in there. So the replication complex may not actually move along the DNA, but it's thought that this complex is going to be the thing that's more sedentary in the cell or more anchored, and that the DNA moves through it. So it's kind of like, if you're looking at a shirt, you could say, okay, you unzip it up and down, or you can hold the zipper and move the shirt through the zipper in there. Here they think that you're holding the zipper and your DNA moves through as you hold on to the tab of the zipper like that. So when you're moving things along at that rate, copying these little base pairs, errors happen in there. Fortunately, they don't happen that often. In completed DNA, there is only 1 in 10 to the 10th errors left in there. So we remove a lot of errors in there. Your initial pairing, though, you're going to have 1 in 10 to the 5th nucleotides will have an error in there. So we've got a lot of mechanisms in there that the DNA polymerase itself is going to proofread as it goes, but sometimes it misses things. So mismatch repair, you have other enzymes that come along as proofreaders in there, and they're going to replace some of the incorrect nucleotides in there. Your DNA, however, is constantly exposed to mutagens, and while most of the errors are usually corrected, after you've gone through and done the proofreading, if that mutagen does their damage once you've done the proofreading in there, you're not going to catch it. So it's kind of like if you're proofreading something on your computer and you go, okay, I have proofread the whole thing, and as you turn around to walk away from your computer, you put your hand on the keyboard and you type in a bunch of letters in there and create some mistakes after you did your proofreading. They're in there. You missed them. And it's the same thing that could happen with the DNA in there. So we know in E. coli, it has almost 100 repair mechanisms. So far, we've identified about 130 of them in humans. Likely, there is a whole lot more out there that we don't know. So an enzyme nuclease is going to be a DNA cutting enzyme in there. What it's going to do when you see that there is a problem is it goes in and it cuts out the damaged DNA, and then it allows that resulting gap in there to get filled in with the new nucleotides. So this is referred to as nucleotide excision repair in there when you have enzymes filling in the gap. So one type of mistake that we'll have are thymine dimers in there, where you have 2 thymines that are next to each other on the strand, and UV light can cause them to bind to each other. Now they're not read as thymine anymore. So when the enzymes are going along reading this, they don't pair it with A, they pair it with other things because it doesn't look like thymine anymore. So evolutionary significance of the altered DNA. Rare mistakes do slip through, and it introduces variation into the gene pool and allows evolution of a diverse species in there. When you introduce in a variation, you may have a variation that is better for the population. You may have one that doesn't make much change, or one that makes one that's worse. To get one that is better than the population is pretty rare. There. Most of the time, if it puts in something different, it's usually non-functional and doesn't work and doesn't get passed on. On the ends of the DNA in there, you can only add to the 3' end. So when you're doing the copying here at the 5' end of this, there's a little bit of it here that you can't really add on to. And so that 5' end on there is a bit of a problem in there. And so we have a little bit of extra stuff that we're going to stick on it. You can't replicate it or repair it since there is only the one end you can add on to in there. So they're going to have telomeres on there, which are special little sequences in the eukaryotic cells that they don't contain genes, but they have repetitions of short little 6 nucleotide sequences in there. And so each time you copy that gene in there, you erode off one of those sequences, because you can copy most of it. So we'll say you've got your strand here and this telomere on the end. You can never really copy the whole thing. So the next time you make it, it still allows you to do that whole thing, but the telomere is a little shorter. Next time you make it, telomere is a little shorter. Next time you make it, a little shorter yet. So, see any potential problem with that? Eventually you use up all the telomere. So it's one of the theories in aging that you can only replicate your DNA so many times, and then it's not going to replicate right because you have no more telomeres on there. So your human telomere is going to have this sequence on it. And you'll have between 100 and 1,000 of them that acts as a buffer zone to protect genes. It's going to prevent staggered DNA because what will happen is when you can't have this on there and you have to start cutting off pieces of the DNA, it's going to lead to cell cycle arrest. It just can't go on anymore in there. So we've got this enzyme telomerase that can catalyze lengthening of the telomeres in the eukaryotic genes and restore their length, but this is not active in most of our cells. And you look at that and you go, okay, that really sucks that that's not active in most of your cells. But it's thought that those telomeres shortening in there is kind of a self-limiting process in some of the cancers. They will only get so far because they can only replicate out of control so many times. And then it runs out of telomere and it stops the cell cycle and it keeps the tumor from getting too big. Well, those nasty little cancer cells have been found to be able to create some telomerase activity in there where they can just keep on going and create more telomeres that will allow them to become immortal and keep copying in there. So when we look at the chromosome, this is going to be your DNA that's packed together with the proteins in there. In a eukaryotic cell, it's going to be in the nucleus. In your bacteria, it's going to be in the nucleoid region. Oh, we are at 9:47. Then let's not start this part here. Let's stop there today.